Okay, we'll hopefully get through the evolution section this afternoon. Probably not the final classification part, but we'll finish that off in the morning anyway. And you've got the slides printed out. The PowerPoint itself, if it's not on Moodle, I will make sure it goes onto Moodle. I've slightly edited it this year, so there are a few things that take me by surprise as I go through, but otherwise it's okay. And what we're doing really is sort of looking at the logical conclusion of our journey over the last few weeks. I mean, for this unit, it is a big unit. You know, we started off with replication of DNA. Don't forget that replication can introduce mutations. We then looked at the division of cells, mitosis and meiosis. Of course, you've got processes like crossing over and all that sort of thing. Can also increase the risk of changes in the code in the DNA. We looked at control of gene expression, DNA technology. We've then gone through the inheritance part of it, monohybrid, dihybrid, and we finished off last week with the population genetics bit about allele frequencies, genotype frequencies, what influences them, and looking at things like the role of mutations, for example, because we dealt with that several times, being the only real source of variation in the genetic code. The idea of migration being important in terms of numbers of organisms in a population. The process of natural selection was mentioned, that you can only apply the Hardy-Weinberg principle if there is no selection and no migration and no mutations and so on. So this is the scenario then. If we have got selection, then we get evolution. And our starting point has to be really a little bit of consideration of the history of thought on evolution, and a warning to some extent not to believe anything too intensely, whatever extreme. All right, extreme views are never good, as we know. But the history of evolutionary thought, as long as people have been able to discuss their ideas, then there have been theories, hypotheses, ideas, whatever you want to call them. You can go back a long, long way in history and look at ideas. Now, you can also, of course, look at what we'd consider to be unscientific ideas about evolution, and you can look at, the idea of creation as it's presented in the Bible, for example, and you get a sequence. You get a sequence that says, right, you have to start off by making planets with light from a sun, and you then start making then water sorts itself out and land and all that, and then you start to get animals, and then eventually you get humans, and then when you think about it, you think, actually, science actually does describe that. And so you have to be, well, apart from being respectful of other people's views, beliefs, and all that sort of thing, you also have to be quite open-minded. I think the worst thing you can do in science is assume that everything is fact, because it quite evidently isn't. There is an awful lot of flexibility. Sometimes we find, just one tiny little new bit of evidence on something that changes everything we thought we knew. And then other times it takes a while to put together, oh, we can add that in, we can add that in, we can add that in, and you start to build a bigger picture. So the first thing I've got is a slide that just suggests some of the theories on the origin of, that should say life at the top. We decided actually it should have said life on Earth this morning. So I really ought to edit that. I've also edited a spelling that's been wrong on this slide for as long as I've been using it. And you'll see on your sheet, it says Cosmozoan with a Z, 3 letters in. And it's been like that for as long as I've used this, and that's a long time. And yeah, nobody'd ever spotted it. I was just looking at the screen this morning and thinking, that just doesn't look right. This is by no means an exhaustive list of people's proposals over time. But, you know, it's not my job to sort out which of these is your particular view. All right, but what you must be wary of is being, you know, too stuck to one particular view without any consideration of any of the others. We'll see why in a minute. And what most people end up doing is sort of knitting together a bit of this and a bit of that and coming up with their own feeling about it. Probably none of which can ever be proved, but hey, you know, that's it. A lot of science will never be proved. So if we start at the top, the idea of special creation, obviously the extreme religious view that, I put supernatural being, but you know what I mean by that, a god if you like, but not necessarily, because if you go back, you know, into sort of earlier religions, you're not talking about a god as such. But this idea that, you know, somebody somewhere, a being if you like, was responsible for the appearance of life, and it's a way of satisfying a need to know, but it's something it's very difficult to prove didn't happen, you know. But you know, you mix it in with the others, and like I say, you come up with an interesting solution to it. Spontaneous generation. Now there's an interesting option. I think we're probably a bit beyond that. Life arose from non-living matter on several occasions. And it's on the several occasions that's the curious bit. All right, well, non-living matter, we're talking chemicals. Well, all right, if we take the biochemical view, then, you know, we would have to say, well, what came first biochemically? You're talking about probably amino acids. What are amino acids made from? Oh, carbon, hydrogen, oxygen, nitrogen. Non-living matter. So, you know, you sort of have to pull these fragments out. And I think, you know, keeping a really broad, open mind, but, you know, balancing it so that you're comfortable with it is really important. Steady state, life has no origin. That's a tricky one. Cosmozoan, life arrived on this planet from elsewhere. How are we on that one? There's the bacteria on asteroids or whatever is actually thought to be a possible, you know, so that's not, you know, it's not that hard to incorporate. And then the biochemical evolution, life arose according to chemical and physical laws. You start to realize, I hope, that when you mix in some of the others, that actually that on its own is equally as dubious as any of the others, really. Just, you know, to assume, well, how? It arose? What do you mean it arose? So, you know, we're not going to have a debate on it, but, you know, do just bear in mind that you can be as flexible as you like in your understanding. But what we're going to look at, of course, is the process, not so much about the origin of life, all right, because that's a question it will be pretty nigh impossible to answer ever, I should think. We're not concerned with origins. We're concerned with the changes that have occurred since. So let's assume we walk in to find life on the planet. We'll work from that, okay? But when I talk about, you know, putting them together, knitting them together to make your own interpretation, it's this sort of thing. This comes out of the same book that the last listed. But the idea of steady state is a tricky one because, you know, if that was the case, then humans would have been as they are, you know, for as long as time goes back, and we are fairly clear that we haven't been. Special creation is a harder one to knit in, but there's room for it. Spontaneous generation is sort of part of that somewhere. I love this bit at the bottom. This is why I've included this. Cosmozoan, yeah, all right, originated somewhere else and then came to the Earth and then developed. Well, yeah, you've then, if you're looking at the origins of life just as the broad thing, well, it had to start somewhere else, so how did it start there? You've got to answer that one. And then the biochemical evolution at specified times, yeah, all right, well, that's actually not very different from spontaneous generation if you think about it. So, you know, it's quite a I would say leap of faith, but that doesn't seem quite right, does it? Yeah, well, spontaneous generation doesn't so much. No, not stone to human, but stone to simple cells. You know, that simple life originated from non-living matter and then developed. Is sort of no. It's the well, you get it sort of down here where you start knitting them together. If you look at this, if you assume that life has come from somewhere else, it could be special creation or cosmozoan is an option. Either of those would imply spontaneous generation, that a zap just deposits it on the earth. From there, you can then go through a biochemical process. So none of these are you know, a big picture. None of them are the full story. But, you know, it's just leaves space for new information in your understanding of this, really. Okay, so who do we have to thank for the way we currently understand biochemical evolution? Good old Charles Darwin. I shouldn't use that picture because that's him as an elderly man, whereas all his work was done when he was young, but yeah, I just do. The handout that it refers to is actually the next 3 sides, I think, that you've got, where you've got a bit of history of understanding, history of ideas. Now, this particular one starts at the 17th century, but you can go way, way, way back, you know, and people have, like I said, as long as they could talk to each other, they've talked about where they came from. So, you know, this is a very short history. Most of it sort of started happening around the 19th century. Mid-1800s was an incredibly busy time in all sorts of senses. You had the Industrial Revolution, and so many things changed in the mid-1800s. But Charles Darwin came out of that period being the most influential person in terms of you know, the way that evolution was understood. But you have to remember, he knew nothing about genetics. But then again, neither did Mendel with his pea breeding experiments. And of course, they weren't even talking to each other. So this idea of inherited characteristics. So, you know, it's actually remarkable that they came up with the ideas they did in total isolation. You also have, in your notes bit there, before Darwin, you've got a guy called Lamarck, who's a little bit earlier than Darwin. His ideas, you know, we tend to sort of say, oh, that's rubbish. But we have to slightly revisit that. Lamarck's ideas were based around the fact that he thought that organisms would change during their lifetime in order to meet the demands of the environment. So the classic is, you know, amongst a group of giraffes, you might have one that stretches its neck more than the others, manages to reach the higher leaves on the trees, has a feeding advantage, a survival advantage, a reproductive advantage, and so with its stretched neck it reproduces and produces offspring with longer necks. That, in essence, doesn't work. Because the logic from that would be, if you had to have a leg amputated in your early life, then any children you had would be missing a leg. So physical changes in your life aren't going to be passed on. Similarly, have a tattoo, children are going to be born with a tattoo. You know, and it doesn't work, does it? That sort of physical change. And it's hard to stretch your neck that far, to be honest, as well. Otherwise, I'd be taller. So, yeah, we sort of, you know, call it rubbish, but we do actually now know, don't we? See, epigenetics being our, you know, newest sort of aspect of understanding, that you can change your DNA in your lifetime. So therefore, if you can change your DNA, you're changing your genes, and what you were born with is not what you pass on to your children. You can change them by diet and by habits like smoking and that sort of thing. So, yeah, you know, I mean, Lamarck didn't actually suggest that sort of thing, but we do now know that he wasn't a million miles away from, you know, some possibilities. However, probably worth a mention more than him, and I mention this because Bill Bailey reminded me a couple of weekends ago, just one of those random Sunday afternoon programs you find when you just can't be bothered to do anything else. Was a documentary that Bill Bailey had made about another guy who was working at the same time as Darwin, called Alfred Russel Wallace. And it was the second time I'd seen it, actually, and I only remembered it sort of when I saw it, and I really should make a bit more of Wallace's work. But he was working at the same time as Darwin. I can't remember where he was, but while Darwin was out on the Beagle, the ship that was mapping South America particularly, Wallace was, I can't remember where, I've completely forgotten. But Darwin made a lot of notes and things on the Galapagos finches and tortoises and that sort of thing. And Wallace was looking at things like birds of paradise at the same time. You know, they were literally doing these things at the same time. And Wallace had written up his ideas, which pretty much were the same as Darwin's. And he wrote to Darwin before he published. Big mistake. Because Darwin realised that Wallace could easily get ahead of him in the publication stakes, and of course in those days it mattered. You know, if you were the first person, your name was attached to it, and that was that, it forever would be yours. And so Darwin really got a move on. He published his book. Origin of Species is what we call it. Its full title is On the Origin of Species by Means of Natural Selection, or the Preservation of Favoured Races in the Struggle for Life. Yeah, I haven't ever got round to reading it fully. I keep starting it, and I've pretty much given up on it because I can't cope with the language of it. It just, unfortunately is not for me. But he managed to publish, and he, I mean, the way they did it was to hold meetings, you know, where they gave sort of lectures, if you like. They presented their research, you know, at, I don't know whether it be the Royal Society or something like that, you know, sort of respected organisations, and he managed to do that before Wallace returned to the country. Although I can't think what Wallace's book was called, but he acknowledged Darwin's, you know, genius and all that sort of thing as well, so very sort of decent about it. But Darwin, of course, you know, really sort of set himself up for criticism and anger, because this was a new idea. You know, really to present this as a scientific idea was sort of seriously groundbreaking, and people didn't like it because religion was very important to people, and it implied that you know, that the creation side of all of that had no part to play, and you know, at that point it wasn't about sort of saying, right, well, you know, okay, God might have created life and then it evolved. You know, people weren't able to do that. And there was quite a lot of sort of backlash against Darwin for suggesting all of this. And there was a phrase that's attributed to him that he didn't actually use, which is, you'll know it, survival of the fittest. Yeah? Yeah, that he never actually said that, I gather. There was some implication that humans evolved from apes or monkeys, and there was a, you know, he was drawn as a cartoon, as a monkey. Yeah, and that was it was actually on last week's GCSE biology paper. Yeah, and it was really just because the implication was there, but he didn't actually say that at the time. And of course people were just misunderstanding because it appeared to attack their beliefs. Of course, you know, they couldn't cope with it. The important thing to understand is that modern apes and monkeys are as evolved as we are, but we have a common ancestor. And this is what they weren't getting, you know, and it was this implication that human beings were no better than monkeys and all that sort of thing. So it took a while for all this to be accepted because there wasn't any other evidence. You know, what Darwin had done while they were mapping, taking this boat round the coast of South America, you know, making measurements, drawing the maps, and he was going onto the land and having a look at the plants and the animals and recording all this. And when they got up the west side to Ecuador, isn't it, where the Galapagos Islands are, he really started to realise that there were, and it was the finches particularly, on the mainland of Ecuador, which had similar looking equivalents on the Galapagos Islands. But on the different islands, there were different types of finch. And the way they varied most, I mean, apart from body size and colours and things like that, was the beaks. They might be long and thin, they might be short and strong, they might be, you know, all sorts of different types of beak. And those seemed to correlate with the sort of food that was available. And yet it looked like they had a common relative on the mainland. And this was where he started making his observations that in any population you have variation. In any population, some individuals survive and some don't. The populations tend to stay about stable. And it's about this constant battle, you know, for survival. I can't remember what the phrase was that was used, actually. But, you know, started realising that there was selection of certain individuals going on, depending on their characteristics and the environment. So this is where all this came from. And so, you know, he started it all off. He didn't have the sort of knowledge that we have now in terms of things like genetics, and you know, he hadn't looked at fossils and a lot of the other biochemical stuff you can do, which we'll come back to in a minute because we've added a lot more evidence since. But this idea of natural selection, you know, this was the big breakthrough. Now, of course, you can never really use humans as a very good illustration in evolution because we tinker with it so much. You know, we are not subject to natural selection much, are we? We are battling natural selection all the time. You know, we don't let our population members die if they are, you know, perhaps weaker for some reason. We help to support them. So you know, we do interfere with it a bit, but, you know, we have a right, maybe. Yeah, we've got we need to look at some different types of selection, and then some of the evidence that goes with it as well, and then look at how that can then lead to the formation of new species. Okay, natural selection is actually only one option. Of course, there is artificial selection as well. And that's something humans are very good at, artificially selecting individuals for certain features, particularly in agriculture and animal breeding and that sort of thing. You know, you look at dogs, for example, all originating from a wolf. Yeah, they don't bear much resemblance anymore, do they? And that's artificial selection. You know, we've decided they should have squashed noses, short legs, ridiculous folds of skin. You know, it just... yeah, when you look at the damage it causes as well, you realise, you know, I think we mentioned it with the idea of inbreeding. But artificial selection, you know, has been important in agriculture for increasing milk yields and low-fat meat and that sort of thing as well, and in plants for increasing yield and, colours in flowers, for example. You know, there's always the hunt for, I don't know, the black rose or something like that, isn't there, you know? Yeah.
I read this thing on foxes over on Twitter.
Yeah. Oh, right, yeah.
There was an article, I think it was in the Scientist.
Right.
He published a paper, and that was the characteristic change. So they got a shorter tail.
Right,
they
yeah.
had an extended adolescence.
Yeah.
And they were like slightly smaller, and it took a certain amount of generations before they kept...
This is the thing, yes. Once you start selecting what you want, then...
yeah, they didn't like publish what
No?
actually happened to like the genes. Like,
right. No, I think a lot of it must be quite subtle, and interactions between lots of genes will be involved sometimes. But, you know, I mean, humans generally, you know, you look at the world around us and we've selected so many options out of it, haven't we? And, yeah, discarded others like purple carrots, although they are making a comeback. We tend to now refer to neo-Darwinism rather than just Darwinism. Darwinism just, you know, doesn't take account of all the additional evidence that we've got. But we've now, you know, we now know, particularly the genetics side of it, you know, that we're not just talking about the selection of certain characteristics or something like that. This idea that we now know what's responsible, that it's the DNA, it's mutations in the DNA that give the variation, and the fact that that's what's being selected actually, which he knew nothing about. Now, where we go from this then is that in order to get natural selection on a population, you've got to have a selection pressure or pressures, and the result of that pressure. All right, so that's sort of the mechanism, if you like, or the cause. The mechanism then, I suppose, is the natural selection, which is that one or more of the genotypes will have a survival advantage. If they've got a survival advantage, that means they can reproduce more. They're more likely to pass their genes on. And so it's this survival and reproduction together. There's no advantage in surviving if the animals or plants don't reproduce. It's got to be that handing on the genes, the survival genes, to the next generation that's the important thing. But this idea of selection pressures, it could be perhaps the colour of an organism in a certain environment that might change colour. It might be temperature. It could be all sorts of different things. So we'll have a look at a few examples. First one then, directional selection. Now there are going to be three types. We'll have directional, stabilising and disruptive. First two of those come up in the exam. So this is the first one, directional selection. It does what it says. Now you should remember the normal distribution from our very first unit from cells when we were doing the data handling bit. The bell-shaped curve, the line down the middle being the mean, and as it shows you here, we represent the mean as x bar. What does the delta mean? Change. So what that little bit, delta x bar, means change in the mean value. All right, it's just a quicker way of writing it. So the change in the mean value in this particular example, it's being pushed to the higher numbers perhaps, or to the right if you like. The mean itself actually moves. The whole bell-shaped curve, the whole normal distribution has been shoved along the x-axis from left to right. So our selection pressure is favouring the higher values of this and not favouring the lower values. So to take a rather, well, it's not a good example, you might have, let's say, fur length in a mammal that perhaps in an environment that suddenly becomes colder, or maybe it moves to an environment that becomes colder for a reason. And it might be that longer fur provides a better insulation, good at keeping the heat in perhaps. And so the animals with the longer fur are better able to survive in the cold, pass their genes on, and so on. You'll always have a tail off because, even with long fur, there's a limit, before you start tripping over it, and then small animals tripping over long fur are going to get picked off by predators. So that's not a good thing. Perhaps a better one would be thinking about colour. And there's a slide coming up that has a nice example of this, where you might have a population of little mouse-type animals where some are a sort of light sandy colour, and then going through all shades of brown until you get to a real dark colour. Now, if their environment changes, let's say, so that we lose some trees where the dark ones might be better camouflaged, then the preferred coat colour would move towards the lighter ones because the dark ones are going to stand out more. If they're in a mixed environment, then they need mixed colours to survive in different areas. The example, however, that is perhaps one we can relate to, given some of the things that we've talked about, that I've got here: antibiotic resistance in bacteria. And what we do, of course, is to put antibiotics into their environment. And if you think of something like penicillin, we did do a bit on this. I think it's come up actually several times. When we were doing cells, and we talked about bacterial cell walls, we talked about the structure of them. And then when we did proteins in the biochemistry unit, part of the PowerPoint on proteins, I showed you the structure of an enzyme called beta-lactamase. Beta-lactamase is an enzyme that some bacteria have, which can break down penicillin, because penicillin, the main chemical structure of penicillin, is a structure called a beta-lactam, which is a ring shape. And beta-lactamase, well, if you're a bacterium with the mutation to produce beta-lactamase, you can break down penicillin, you survive. Right? And this applies to other chemicals used as antibiotics as well. There'll be enzymes that can break down their basic chemical structure. So if you put loads of penicillin into the environment, then you will almost undoubtedly have at least one bacterium in the population that can produce the beta-lactamase. So it survives. And, you know, if you allow this to go on generation after generation after generation, you get lots and lots more of them. And so gradually the tolerance, or the resistance, if you like, is moving, as it is on here, from potentially 0, but with a mean at low numbers, to much higher levels. So the resistance increases, all right? And that's, you know, an important aspect of understanding where we get antibiotic resistance from. Now, a second type of selection, which doesn't move the mean value this time, it's stabilizing selection. With the directional one, you weren't actually changing the normal distribution, just moving the whole thing along the x-axis. With this one, you are not moving the mean, but you are removing the extreme values. Now, this might be, if we considered our mammal with the fur of different lengths, it might have to move to a particular environment, and then over time you lose perhaps the shorter fur and the very long fur, and it just sort of stabilizes out. Similarly, with the color option, it might be that you know, very dark fur and very light fur are both, you know, a bit more of a disadvantage, and actually medium colored fur is probably your best survival if you're in perhaps mixed plants and things like that. And what we get here, the delta again means change. VAR, we haven't actually used that, but it's variance. What measure did we use for the spread? No, when we did that unit, you had to work out the mean and the standard deviation. Yes, remember the standard deviation? That's the spread either side of the mean, and it's sort of, I think, like the square root of the variance or something like that. So we are also changing the standard deviation here, aren't we? Because that was the spread either side of the mean. If you've got a large standard deviation, then, you know, you've got a relatively flattened distribution, and the tails go off to, you know, quite a long way along the x-axis. The smaller the standard deviation, the closer the results are clustered around the mean. And that's what you were looking for with your osmosis results, is that for each set of results, you wanted the smallest standard deviation possible to show that, you know, between the different people in the group, you know, you were coming up with results that were close to the mean for the group, not that, you know, it was taking, you know, high readings and low readings and just getting a mean that didn't mean anything. So with this then, what's happening is our selection pressure this time is sort of pulling the sides in. The mean hasn't changed with this one, it's just that you've got fewer at the low end and fewer at the high end. Right? Most are clustered around the mean. Now, the example that I've given you for this one, this is a classic, human birth weights. The blue part, which is a histogram, I believe, a shape made out of sort of like a bar chart with no gaps in it, shows birth weight in pounds. So that's down the left-hand y-axis for the percent. No, hang on a minute. No, that's our percentage. It's on the x-axis. And then we have the percentage of births in the population up the left-hand y-axis, sorry, so that's recording that. And then up the right-hand y-axis, whatever sort of scale is that? It's logarithmic, yes. It doesn't look obviously so, does it? But what you've got here is 10 to the power 1. What you've got here is 10 to the power 2. So, of course, it's a way of getting quite a big spread of results onto an axis. Quite tricky to plot, and I don't think we've come across... You haven't used logarithmic graph paper in maths, have you? You'll be grateful for that. Logarithmic graph paper is weird stuff. It's not squares as such. The scale is, I can't remember whether it starts with the big ones and gets smaller, but yeah. So if we were to make the next division of that distance up this, all right, the next division up the board of that size would be, what would we mark off? Sorry, 1,000? Yeah, 1,000 would be the next one. 10 to the power 3 is the next one up. So it means that you can have 10, 100, 1,000 on the same scale. Tricky to read, but you know, that's it depends what sort of results you're handling. I think we could have used it in chemistry actually in the rates bit. Because if we'd done the log of the rate against concentration for the, I think one of them should give you a straight line instead of a curve anyway. Right, back to this. So we would associate the lowest birth weights with poor survival, yeah, and yeah, prematurity, and the highest birth weights also with low survival. But the middling birth weights give us the best survival. All right. Now, over time, this has had an effect, and the majority of human babies are somewhere between about sort of 5 and 9. I think that's slightly off actually, but yeah, between about, say, about 5 and 8.5 pounds. I have a slightly distorted view of it from our family, maybe. Yeah, although I don't know. I mean, ours tend to do all their growing after they're born. They're not huge, but they become huge. Interestingly, the sort of standard is that newborn babies lose weight in the first week and then regain their birth weight in the second week. None of ours have ever lost any weight at all. They just pile it on £1 pound a week. So by 2 weeks they're £2 heavier than they were born. You literally, yeah, it wasn't just my two, but my grandsons are doing the same thing. Yeah, so like I say, I have a slightly distorted view, but you know, ours have been around the sort of 7 to 8, well, 8.5 perhaps. But you know, why have we got this sort of correlation? And again, you know, you've got to be careful the correlation causation thing, that one doesn't imply the other. But there are links, aren't there, that we know low birth weight babies are very often premature, so they're not fully developed, so they can't support themselves particularly well, and their lungs may well be underdeveloped. That's usually the big problem. What about the high birth weight babies, then? Why aren't they? Yeah, it can be. Yeah, gestational diabetes tends to produce large babies, but why don't they survive so well? Yeah. Well, it's not really obesity. You know, they can just be big. The babies are just big. Yeah. There's yes, there's that sort of thing. So yes, if they are particularly big, then birth itself can be more difficult because yes, they won't fit through the pelvis necessarily, so they can be more traumatized by cesarean, perhaps. Yeah.
Such a long she's all not breathing.
Yes, taking longer to be born can put them at risk. The other thing, though, is that they can be too big for their systems, as it were. So things like maintaining body temperature can be harder because they're bigger than their developmental age would, expect them to be, yes. And, even things like controlling water levels and sugar levels and all that sort of thing is harder if you are bigger than you should be for that gestational age. So there are disadvantages to small babies for obvious reasons, large babies for perhaps less obvious reasons, but you work that out, okay? So what's tended to happen over time is not that babies are getting bigger and bigger. That is not an advantage. Definitely for the mothers, it's not. Anyway, but that we've had this stabilizing selection, essentially, that small, particularly small and particularly large, are less likely to survive. So, we end up with losing the extremes, losing the very light ones, the very heavy ones, and, stabilizing around the middle. Of course, if you've got a high-tech population, then we tend not to lose them. Not so much now, do we? And that's what I mean about human beings being a really bad example of evolution. That we're interfering with evolution. I mean, yeah, okay, I'm not saying we shouldn't. That would be, perhaps not appropriate. But, by using technology to support very premature babies or very large babies, we are avoiding the selection process, aren't we? All right. So we've got the directional selection moves the whole distribution, including the mean, in one direction. It could be the other way. Stabilizing selection takes off the extremes and favors the mean, really. And then we have, this one is a bit odder, and I'm not going to give you an example of this one on the exam paper, but we need it here really just for completion. And this one's called either disruptive or diversifying selection. And it can go as far as resulting in new species. So if we've got the same old blue distribution there, our normal distribution at the start, what happens here is not that we get a push in one direction, pushing it one way, not that we get a push compressing the sides and taking off the extremes, but we get some sort of selection pressure actually from inside the distribution, so that the mean value is no longer favourable. And this is, so it's an interesting option. An example I was used to, I can't remember where I got it from, but an example I was used to use for this was cattle with and without horns. And I think it must be a particular breed of cattle where you either had them with or without. There wasn't a whole range of different horn lengths. They either had horns or they didn't have horns. And it wasn't that you could have them, any length from sort of, stumpy to long or whatever. But that's not something we're perhaps very familiar with. But what happens here is that we again get a change in the variance, the spread, if you like. This time it's increasing. We're moving away from the mean and actually broadening the range of genotypes here, or phenotypes actually. And what we end up with could be, down at the bottom there, extreme disruptive selection, where you appear to have, two humps. It's a bit camel-like. But we could actually lose that bit in the middle there. It could actually end up going down like that. So we end up with two completely separate populations. Now, if those populations then become genetically different enough from each other, you've got two new species, and you lost your original one. So, that was not unheard of. You've got an example now on the next slide that, unfortunately, because they never copy particularly well, but it looks much better on here, that actually shows all three options. If we take these little, I don't know what they are, rodent-y things by the looks of them, with a range of the colours from very light through middling to very dark in our original population there, so that we've got a, you know, quite a spread, but giving us an original population with a nice, clear, normal distribution. A few very light ones, a few very dark ones, most around the middle. If we were to apply our selection pressures in the different ways to that little population, then along the bottom there, the first one on the left, you've got directional selection. This would suggest perhaps that these animals had moved into perhaps an area that had some small trees, and gradually over time the trees got bigger and filled out and produced more cover, more canopy, and produced more shade, and so more dark areas. Now, what you would get as the forest, as it would become, got darker and darker underneath the trees, would be a selection pressure that favoured the animals with the darker fur. More shadow, more dark places to hide, better camouflage. So the dark ones would be better at surviving. The light ones would be more likely to be picked off as prey. All right, so you'd have predators coming along, being able to see the light ones in the shadows, and you'd lose those. So your population would evolve by becoming darker. All right, now this doesn't just happen over one population or two. You know, this is a long-term change that we get. The middle one, well, we might have had an area, they might have been living in an area of mixed shelter, you know, perhaps some open areas, some bushy bits, some grassland, some trees, where most of the animals had, you know, some chance of survival and shelter, being camouflaged in perhaps different parts of where they were living. But let's say some change happened to that environment where you were left with just a forested area and then some, I don't know, sandy open ground, that the area with the light shade disappeared. What you then get is that the animals with the lighter coats, of course, are favoured in the sandy areas. You know, you think about being down on the beach this morning, a little sandy coloured animal running across that wouldn't be so easy to see. The darker ones would, so, you know, being sandy coloured would be an advantage. On the other hand, you know, if you've gone up to the cliffs and into the woods up there, then the sandy ones would probably stand out. So the darker ones are more favoured. The ones in the middle probably show up in both environments, so they're a bit stuck either way. And so you tend to get this diversifying selection where the overall favour is in terms of the extremes rather than the mean value. And then, of course, the opposite to that is the stabilising selection. But that tends to happen once you've got the other ones. Once directional selection's happened, or diversifying selection, and you've got your populations sort of settled where they are, you then tend to get the stabilising, taking off the extremes. So, you know, this might be an example of, it's not perhaps a good one here. I think we should have had just a dark mouse or just a light-coloured mouse. But we lose the extremes. We've lost the light ones, the dark ones. We've been left with the middle sort of shaded ones, suggesting that, yeah, right, we've lost the trees and the sandy area has got something growing on it now. We've got this middling sort of light shade, and the middling coloured mouse is favoured. It's not a mouse, is it? Animal, mousy thing, I usually call them. And so we've got, you know, different mechanisms occurring in these different examples, but they all result in the same process of change, you know, genetic change. Change in the genotype frequency, which of course was what we didn't have with Hardy-Weinberg. Right, now let's have a look at the evidence bit of it then. Okay, well, Darwin came up with observations and descriptions, and probably the best part, best sort of one of these to compare it with would be the comparative anatomy. So he looked at the finches on the mainland, he looked at the finches on the islands and compared them. Now, the phrase I've used in here sort of summarises evolution in one sentence. by using a number of bits of terminology. So I'll do that first and then go on to the other ones. This idea of adaptive radiation, it's sort of back to front, because you get the radiation first, then the adaptation. Now, radiation means that if you've got all of your organisms in one place, they then radiate out away from that for some reason. Reasons we shall come back to. But they could all be in one area, let's say, that suddenly suffers a drought. So they have to move away, and some go north, some go south, some go east, some go west. And so they radiate away from their original environment. And each group, you know, the north, south, east, west groups, each one encounters a different environment. And in those different environments, some will be suited to survival there, others won't. So you might have some go north and meet a forest where, you know, yeah, I'm not using the Galapagos finches here, I'm still with the mousy things. If some went north and met a forest, the dark ones would be favoured. If some went south and met a beach, then the sandy ones would be favoured, and so on. So they then, over a number of generations, adapt. So you get more and more showing characteristics that suit them to that environment. So that's adaptive radiation. It can go as far as, if we think about the finches, the one thing that Darwin looked at mostly was their beaks. And the beaks of all birds are what we describe as homologous. Right? Homo means same. Homologous, we've used it as a word for chromosomes in a pair as well, haven't we? You know, matching ones. Homologous characteristics would be things like a human arm, a bat's wing, a seal's flipper. They're homologous. They've all got one bone, two bones, a lot of little bones with a pattern of 5 at the end. All right, they're homologous. They've got the same origins, but they don't necessarily look the same. And the way this occurs is by a process that we call divergent evolution. So you start with one original beak or limb, and it diverges, it moves away from the original, but the pattern is still there. So they've got this one common ancestor, if you like. You can also get convergent evolution, where you've got different origins, but characteristics turn out looking the same. So, for example, birds' wings and bats' wings are homologous, because they've got the bones in them, in the bone pattern. But bird wings and insect wings are analogous. All right? Homologous means with the same origins. Analogous means sort of comparable, but with different origins. There was another one we did. Actually, I mentioned it briefly, I think, when we were doing eyes. Human eyes have a blind spot, don't they? Well, vertebrate eyes have a blind spot. Can you remember why? What leaves the retina at that point? Optic nerve. And the reason for that is that in a human, well, in a vertebrate eye, if that's the retina, the nerve fibres come out over the surface of the inside, and they have to go out like that. So you have all those nerve fibres coming out and actually giving you your blind spot here. If, however, you look at the eyes of, I think it's octopus eyes or squid eyes, they'll be the same. If you look at their eyes, again, retina, but the nerve fibres come out at the back. All right, so the nerve is actually, it's a more sensible design, actually, all right, but they come out underneath. They don't have to dive through the retina to get out. But the effect, otherwise, they're the same. They've got, you know, liquid-filled chambers. They've got a lens. The structure is almost identical. But they've not come from the same origins. They are analogous. All right? Squid eye, octopus eyes, I think it is, and vertebrate eyes. Whereas homologous would be things like bats' wings, birds' wings. Right, so you can make comparisons like this. Now, I'm not sure what I was doing with wings and eyes, but other bits of evidence that Darwin didn't have, Wallace didn't have, Lamarck didn't have. Biogeography, I suppose they did. Just looking at where they were, and you know that's what Darwin was looking at, wasn't he? Comparing these finches with what's on the mainland. But paleontology, yeah, look at fossils, but until you've got a lot of them where you can start building a whole picture, you know, they don't mean anything in isolation. And then, of course, radiocarbon dating. What isotope of carbon is used? 14, yes. Carbon-14. This works on the theory, which I assume has, you know, validity, that the proportion of carbon-14 in the atmosphere has always been the same. So if you look at the amount of carbon-14 in organic, you know, preserved organic material, or anything, because somebody this morning said, what about ice cores? They do it with ice cores because they have air trapped in them, don't they? But if you look at the proportion of carbon-14 in there, then you can work out, you know, from the process of radioactive decay, because you know the half-life of carbon-14, you can work out how old it is. So, you know, that contributes as well. What else we got? Oh, there's the finches. Yeah. Yeah, quite a lot of them. You see, the one down at the bottom, the common ancestor from the mainland, this is what Darwin started with, and then put together, you know, a comparison of quite a variety that you wouldn't think were necessarily linked, would you? You know, they look quite different, but they are actually all finches. I have no idea what the definition of a finch is. And it depends on the food source on that particular island. I was going to say planet. On that particular island, you will have certain food sources, which might be seeds, it might be insects, it might be flowers, buds might be available, and the beaks are related to the to the type of food that's available. So yeah, that was riveting. Yeah, birds' beaks I don't find that interesting, but because it's a classic piece of evolutionary study, you know, you just have to always say, oh, like the Galapagos finches or Darwin's finches. Yes. Yeah, the important thing to get from this really, this is what you'd call an evolutionary tree, and it's a matter of identifying sort of common ancestors, really. Anyway, other bits of evidence. This is an interesting one. It's a very old image, this one. Hence the drawings look a little bit odd down the bottom. But across the top, you've got 6 embryos. And I should do this by a reveal, shouldn't I? I should do sort of 3 sequential slides with a bit more and a bit more. You know, as if I just showed the top bit first and said, you know, do you think they're different organisms? You'd say, not very different. You know, they do look different, but they look like they're probably closely related. And then the second one, you start to think, oh, right, yeah, there's a bit of difference starting to show here. These 2 on the end look a bit sort of fishy or perhaps reptile because that's got, you know, legs starting to show. And otherwise, these 4 look pretty similar. You know, they're getting limbs. And then, whoops, and then you come down to the last one and you realise, you know, actually they all started off looking the same, and what the assumption is really is that it's almost like embryology is repeating evolution. There is actually a phrase, a quote that I'm using, oh, I can't remember what the words are, but using, yeah, not everyday words, like embryology recapitulates ontogeny or something like that. Oh yeah, I've probably got that wrong, but just think of it as embryology is repeating evolution. You know, that those vertebrate embryos they are up there, or chordate embryos, they've all got a spinal cord. You know, I mean, that's what you can see. You know, that's what this bit is, you know, in all of them. And they've got what would well, these things here, I think, become gill slits. So they are then gill slits there in our fish. They become external gills in our salamander, that is. They disappear inside in other animals. But then the further you go down the down the embryological path, the more different they become. And obviously, there's a lot further to go from what we got at the bottom, because, they're not quite like that at birth, but, you've got the difference becoming more and more obvious, haven't you? So studying embryology can tell us something about evolution, because in our embryological development, we have gills and a tail. Now, why would we, if that wasn't part of our ancient history, in terms of development? So, yeah. I'm going to just do... so look, yeah, possibly... I'm not going to go right the way through this, because I can see people are actually sort of quite... Yeah. Had a long day, haven't you? yeah, and you're more active than you realise as well. So having been out, So I'll get to a stopping point and then I'll leave it. We've got plenty of time tomorrow morning to finish this off. I just get to a tidy place. Let me just move that. This bit just needs a bit more explanation, and then I may well stop after this, and then we've got just some examples of natural selection to do that will lead me nicely into speciation and classification. So I'll just explain through this bit, and then I think we'll stop. Comparative physiology and biochemistry. Well, we can look at DNA and proteins and see if there are differences or similarities between different organisms, and that can help us to work out relationships. The thing is, we sort of mislead you a little bit into saying, that a protein will always have the same primary structure. Insulin will always be this sequence of amino acids. Well, it's not. In different animals, it's different. Because, for example, humans used to use pig insulin if they had diabetes, which has the same effect in the human body, but it's not quite the same sequence of amino acids. There's a slight difference, and so on. So you can look at comparable proteins and see how closely related they are. You can look at the DNA sequences, and you get that classic quote, oh, we're 99% genetically the same as a chimpanzee, but we're also 50% the same as a banana. Yeah. And it's because, an awful lot of the DNA is really just about building cells, and, human beings and chimpanzees and bananas are all built of cells. So that basic metabolism, the idea of respiration and protein synthesis and all that, is the same. But this other one, this immunology tests, it's a curious one, but it's like sort of setting up a reference value. It's a bit like the hydrogen electrode in the chemistry. What you do is you start off by injecting human cells into a rabbit. The rabbit will respond with an immune response like we would if you were to have the wrong type of blood or somebody else's kidney and it didn't match. You would mount an immune response to it. You'd produce antibodies, white blood cells, and they'd all go in on the attack and try and destroy whatever it was. So if you inject human cells into a rabbit, the same thing will happen. The rabbit will produce an immune response. If you then add animal serum to sensitized rabbit serum, because at this point the rabbit has reacted and produced anti-human antibodies, you get different results. And this can help you to see how closely related other animals are to the human in terms of referencing them to the rabbit. So this is what I mean. It's like the hydrogen electrode in chemistry. Our rabbit is our hydrogen electrode. All right, so if we say the rabbit responds 100% to humans, we can now make comparisons with other animals. So if you make a comparison with chimpanzees, the rabbit responds 97% as much to the chimpanzee as it does to the human when it's been sensitized to humans. So there is that degree of similarity in the sort of immunology side of it. 92% to gorillas, 75% to baboons, and only 8% to the pig. Well, you know, that's not surprising, really. But it then does make you wonder about the logic of researching the use of pig organs for humans. But it's not quite the same sort of thing. You have to genetically modify the pigs so that they express human antigens. The problem is not expressing pigginess. That's the sort of thing that they're working on at the moment. But yeah, so, you know, these are the sorts of things that Darwin and Wallace and people at that time could only have dreamed of. But, you know, the evidence that we have, you know, when you look at the embryology, the DNA sequencing, immunology, all that sort of thing, it adds so much more to this idea of evolutionary relationships. Okay, I'm going to stop at that point and let you all go and lie down somewhere for a bit. And we've got plenty of time tomorrow morning. So what I'll do tomorrow is to pick up on the examples of natural selection, and that'll take us into speciation and classification to finish that bit off.